Hello everyone and welcome back to the fourth session, week 8 of our NPTEL course in Herbal Product Technology and Regulatory Sciences. So far in the previous sessions, we have understood the importance of pharmacopoeias and we have gone through what are called as WHO guidelines. In this session today, we are going to go through two different set of guidelines, and that are your ICH guidelines and AYUSH guidelines. So let's see what both of these are. ICH, it is basically designed as a tripartite system by the main nations including Japan, US and the European Union. So all of these countries in the European Union, America as well as Japan met in and decided that we need to harmonize all the quality control guidelines, because if every country has its own different set of guidelines, there are a lot of pros and cons, and that's the result it becomes difficult to manage it, and they harmonized what is called as quality guidelines. So ICH was born out of it and founded about 35 years ago, and now it is registered as per Swiss law 2015 as a non-profit legal entity. Now if you go to see, more countries went on being added to it, and so far ICH has 25 members, 41 observers, and it is made up of a working group which has more than about 700 experts globally, which contribute to development of this quality guidelines. So what happens here is, when you develop this quality guidelines, you have to ensure that all the countries have a very uniform criteria about the quality control parameters, and through ICH, the central body, all the experts contributed, given their own contributions, their own country's regulations, put forward and found out a in-between route for quality control parameters, and this is what is harmonization all about. So if you go to see, the intention of ICH is to lay down technical requirements, and this technical requirement is for all the grades of pharmaceuticals which are mostly for human use. So you bring in regulatory authorities, the people who design pharmacopoeias, you bring in industry, people who manufacture these pharmaceuticals, they discuss in different scientific and technical aspects of this product development, that is the pharmaceutical development, and bring about this different guidelines. So the intention of ICH is very much to harmonize the requirements and to ensure a very safe or to ensure a development and registration of a very safe, effective and a very high quality medicines. So in order to do that, a set of technical guidelines were developed, and this technical guidelines were again developed through a lot of pro and feedback, and what you see, through a scientific consensus and with public consultation, these guidelines continue to exist. So if you go to see ICH guidelines, the ICH guidelines are mainly laid down into 4 domains. They are the quality guidelines, which ensure the quality, the safety guidelines, which ensure the safety of products, efficacy guidelines to check the therapeutic activity, and mixed guidelines, which includes registrations and other things. So what categories or what are the differences between these 4 guidelines? So when it comes to quality guidelines, you see, the quality is assessed. So in terms of to assess quality of a product, in this quality guidelines, you will come across something like your stability studies, how much impurities should be allowed, permissible, non-permissible, and what about the good manufacturing practices and risk management is all seen under quality guidelines. Whereas if you want to see the safety guidelines, it is seen that how safe it is to administer this. So a potential risk, like is it cancer causing, is it a carcinogen, is it going to give me genotoxicity, so are the next generations going to be affected for it, is it going to affect the fetus, what we call it as your reprotoxicity, even effects on heart. With recent COVID vaccines coming out, numerous reports are there that maybe it's safe, unsafe. So, one of the cases is also its effect on heart, and for safety you need to do a lot of clinical data. Now, with the advent of newer technologies, ICH also has introduced a non-clinical way, a non-clinical breakthrough for assessing, something like a cardiac ailment such as your QT interval prolongation liability, as well as, other conditions which are causing drug withdrawals over a period of time. So, they are approved by clinical trials, but later on, because the clinical trials weren't, sufficient enough to give in that kind of data, later on these medicines have to be recalled. So, a non-clinical method is also being developed. Now, moving on to next set of guidelines, those are your efficacy guidelines. So, in efficacy guidelines, you see how efficacious your drug is. That means if it is designed to give you a cardioactive effect, how much cardioactive effect does it actually give? So, it gives you a very good detail about design of clinical trials, how do you conduct clinical trials, safety of the people undergoing clinical trials, as well as reporting of the data such as your adverse drug reactions. So, it covers not only the basic categories, also the novel drug delivery systems, biosimilars, modern day medications, antigens, antibodies, vaccines, and so on. So, our efficacy guidelines are very exorbitant, and then whatever doesn't feature in these three guidelines are often brought into the fourth category, which are called as mixed guidelines. So, these are like little cross-cutting topics which do not fit into above three, and this includes your medical terminologies or such as your MEDRA, common laydown or the format of common technical documents, as well as development of standards such as electronic standards, especially for the transfer of regulatory information. So, we will just have a quick look at this website. So, if you go to see here, this is how your ICH guidelines or the ICH website looks like, where you have quality, safety, efficacy and multidisciplinary guidelines. So, when you click in any of this, you get details of all these guidelines. So, you can access every type of guidelines within the portal itself. So, you can see here under quality guidelines, you have Q1, which stands for stability, analytical validation, impurities, pharmacopoeias, biotech products, specifications in terms of impurities, GMP, that is good manufacturing practices, risk management, product development, quality systems, life cycle, and you have like 14 guidelines under quality guidelines. If you go back to see the next one, and that is your safety guidelines. We said safety is more for your toxicity data. So, here you can see every guideline, how do you carry out carcinogenicity studies, genotoxicity studies, all those different toxicity, especially your immuno, pediatric safety and other considerations. So, you have about 13 guidelines in this domain. Going to efficacy guidelines, these are more about your clinical trials, like I mentioned. How do you design, what is the content, what is the special population criteria, what are the general considerations, all of this are brought in about almost 23 guidelines which come under the category of your efficacy. Moving on to the next set, multidisciplinary. You have MEDRA, like I said, electronic transfers, especially for the regulatory information, common technical documents, gene therapy and other miscellaneous studies such as your drug interaction studies, which we did in the earlier units, bioequivalence, all of this will be featured in the miscellaneous or multidisciplinary character under multidisciplinary category. Now, moving on to the main point, and that is about the herbal drugs. We have a special note on herbal drugs, especially with regards to stability assessment. So, if you go to see as per ICH guidelines, when you want to assess herbal products, especially finished product, you to ensure that at least 2 batches out of every 3 batches have been assessed or are being assessed for their stability. So, like you have to take like a 3-month testing point, especially at 30 degrees Celsius, about 65% relative humidity, and if it is like a product, but if it is a herbal drug substance, only one thing that you have to do is measure its stability at 25 degrees Celsius at 60% relative humidity. Now this ICH guidelines clearly says that there is no requirement for doing what is called as intermediate or accelerated stability testing as far as ICH guidelines is concerned. Now here when you want to do the stability studies, how do you carry out the stability studies? The ICH says that you have to do the stability studies especially with respect to the marker substances which can be quantified very well. Now here you can do marker substances alone or you can analyze the whole fingerprint. When you are doing it for the marker substances, you can say there is a good permissible window which is allowed. Now depending upon the substances, depending upon the potency, two categories are allowed. So in case of very potent substances, SA plus minus 5, that is if say for example my potent substance of a herbal product is something like a digitoxin. So if it is 100 milligram, it can only be like 105 to 95. So plus minus 5. And in some cases where there is a huge variation geographically among the species, in that case it is permissible to plus minus 10. That means if my senocides are about 100%, it could be 90% to 110%. So the window is slightly broadened for especially plant materials which show a huge range of variation. But you have to see it critically if the therapeutic activity is also influenced because of this. If the assay value comes high and low or high or low, and then you have to kind of analyze it and you come to know that its stability is decreasing and it's the drug substance is variable, then you have to check whether or not you have to put it in a 10% window or a 5% window. So that specificity is not clearly given and it is left to the discretion of the manufacturers. So this is little ambiguity with ICH guidelines. So in the Indian context, we have one more set of guidelines and those are AYUSH guidelines. So this AYUSH guidelines as are per Central Council of Research in Ayurvedic Systems. The CCRAS lays down these guidelines for AYUSH formulations. So if you see the guidelines, it is divided into two categories. One, a standardization of raw materials which includes testing for development of a standardization of quality parameters especially for raw materials. Then you have SOPs for drug development. They have given a process flow chart for drug development in the guidelines as well as a proper extraction technique for herbal drugs. Now when it comes to the second part of the guidelines deals with shelf life of AYUSH products. Now this includes shelf life or what you call it as date of expiry of Ayurvedic, Siddha, Unani medicines as well as guidelines for shelf life testing and determination of new products. So for some cases like your churnas, ghitas which are already there in the traditional text, they have already given a shelf life. Say for example your churnams may have a shelf life of 1 year. For example, your churnams may have an shelf life of 2 years. Depending upon the ingredients in that particular dosage form, you have a shelf life which is already been given for by the CCRAS for certain category of products. So if you go to see the first part, that is your standardization of raw drug. The standardization of raw drug, the guidelines match very much to the guidelines. Those have been laid down by WHO. Those have been laid down as per the Indian Pharmacopoeia as well. So when I want to do standardization, if you recollect your previous week, we did what is called as authentication, which includes macroscopic, microscopic authentication, foreign organic matter, that is absence of any extraneous matter, organoleptic characteristics which includes color, odor, taste as well as certain morphological characteristics which can also be evaluated by microscope or which can be evaluated by simply tasting or taking a texture of the drug. This also includes loss on drying. Loss on drying is cumulative of your moisture content as well as in some cases the volatiles as well. The ASH values. We studied ASH values as those containing, or basically the ASH values determine what is called as the mineral and the metal content. So if you are seeing mineral and the metal content, you come to know, you get what is called as total ASH, which is a summation of physiological ASH. That means the minerals present in the plants as well as non-physiological ash, which includes something like your dirt, grime or silica minerals which have been added extraneously, and acid insoluble ash, which is mostly dealing with your insoluble matters. Say for example, your physiological minerals, calcium, magnesium, sodium, they dissolve when they are boiled in acid, but your silica, dirt, grime, they don't dissolve. So what you say as acid insoluble matter is mostly your extraneous matter. We also did what are called as extractive values. Extractive value is an indicator of how much amount of substance actually dissolves in your solvent. Now that solvent can be water or that solvent can be alcohol. Say for example, you are dealing with a carbohydrate rich drug such as acacia, tragacanth, you might get what is called as water soluble extractive value, which is very high because carbohydrates are water soluble. But if you do alcohol soluble extractive of acacia, your carbohydrates are not alcohol soluble. So the value will come down to be very less. So the limits have been specified for each and every drug, as we saw in WHO guidelines, as we see in the Indian Pharmacopoeia, and this is even true for your CCRAS guidelines. Similarly, post that you also see impurities. It is seen that Indian products, especially the AYUSH medicines, have lot of issues with pertaining to metal impurities. So CCRAS lays down a limit on metal impurities that should not be present above a particular concentration. See, metal impurities are inevitable. It's not possible to completely get rid of it, but at the same time we should ensure that those are minimal. So take for example, lead is something that we don't want because it is known to cause poisoning. So lead in your AYUSH products should not exceed 10 ppm, your cadmium just like 0.3 ppm, mercury, which is very toxic, should not exceed 1 ppm, and arsenic should not exceed 3 ppm. If these values exceed those, that means your preparations are unfit and most likely they will not be even what you call approved for export. Similarly, you have more parameters which also include your microbial load that we saw. It is said that when you do a total plate count, the total plate count should not be more than 10 raised to 5 per gram of sample if you are taking it orally, and 10 raised to 7 per gram of sample if you are applying it topically, like your lip or your any lotions that you are preparing. Those should not have more than 10 raised to 7 total plate count per gram. Similarly, when you are seeing pathogenic organisms, few pathogenic organisms have been listed by CCRAS, and that includes your Staphylococcus aureus, Pseudomonas aeruginosa, E. coli as well as Salmonella, which is also known to cause typhoid, and this organism should completely be absent because they might compromise a patient who is already ill and they are opportunistic. That means if the person is already ill, they will feed on it, they will grow and they will cause their own disease conditions, leading to what is called as co-infections. Similarly, if you see your yeast and molds, especially the fungus, the fungal count should not be more than 1000 per gram. Similarly, there are categories which are called as mucotoxins. Mucotoxins are the ones which are secreted by fungus again, especially the Aspergillus species secrete what are called as aflatoxins. And aflatoxins are known to cause food poisonings in human as well as in cattle. Now imagine if you are going to concentrate it, the aflatoxin concentration, or if you are going to extract it, the aflatoxin concentration would go really high. So in that case, it says, depending upon the drug, for different drugs, different limits of aflatoxin have been laid down. But if you go to see the pharmacopoeial limit, we say that your aflatoxin B1 which is the strongest and most potent of aflatoxin, which is also considered as carcinogen, should not be greater than 2 parts per billion. Earlier for the bacterial count it was 2 parts per million. Now we are dealing with something like 2 parts per billion. Similarly, a summation of all the aflatoxins, that is your B1, B2, G1, G2, should never exceed 5 parts per billion. The analysis of your remaining sample with respect to active is very important. So once all your quality attributes have been laid down, you have to see how much amount of phytochemicals or active compounds are actually there. That you can check qualitatively when you do a little TLC or chemical test, but there is something which is called as assay, and assay is the quantitative determination of active compounds. So this assays as per CCRAS, as per the pharmacopoeial limits you see, have been done by different methods, which includes your UV, but nowadays modern methods such as chromatography, especially your HPLC, that is high performance liquid chromatography, HPTLC, which is high performance thin layer chromatography, as well as GC, which is your gas chromatography, have been used. So this is something which is pivotal and very vital, and unless and until this all are met, you cannot ensure quality. Similarly, your AYUSH lays down the guidelines for amount of pesticides, especially your organophosphates and organochlorides that have been present, again depending upon the drug substances. Now we move on to the next criteria, and that is the shelf life and the stability guidelines. So shelf life and stability guidelines, when you are doing, as I said, if the formulation is already reported in market, especially your churnas, take for example your Triphala churna. In that case, what happens is the guidelines and the shelf life of it is already carried out for Triphala churna. So you just need to copy or report the same thing. But if you are developed a new product, the guidelines for that says that you should conduct it at least on 3 primary batches. So whatever first 3 batches of your products come in, and now this 3 batches can be a pilot scale. But when you are doing pilot scale, you have to ensure that your pilot scale is not just, like few milligram or few gram. If your actual intended capacity is 10 tons, at least your pilot scale should be 1 ton. That means at least 1/10 of the actual batch size. Only in that case it carries relevance. So it should be carried in almost using same route, same method of manufacture, as well as same procedure that you are going to follow the final process. So same temperature, same pH, same processes, ensure same stability. So you have to reproduce the whole batch that you are going to take on 10-ton scale on a 1-ton scale, and then you are going to pick up 3 batches from that. At the same time, you have to ensure that when you pick up the product, even at 1-ton scale, you have to use the same primary and secondary packaging. You should not change the packaging, because again the stability depends on the packaging also, how it is exposed to air, moisture, so that affects the product, and as a result you to ensure the same containers and closures, both primary as well as secondary packaging. Now this test, when you carry out in herbs or when you carry out in allopathic, we just see the assay. Here you have to do lot of analysis, because these are herbs, they might change color. If they may not change in active, they are still changing color just because of chlorophyll. They might change in taste, they might show fungal growth. So when you are testing, just don't check the active content, check all the attributes we just saw in standardization. So check the physical attributes like appearance, check the chemical attributes like TLC, biological attributes like efficacy studies, and microbiological attributes, that is your fungal and mold growth as well. And once you are done with that, and once all of this are shown consistency, you have to see that whenever you analyze your active, that active should be analyzed using a well validated analytical procedure. It should not be any procedure which itself would show variation in the results. So only a well validated reproducible procedure is what we use for your stability studies. Now if you go to see what would be the testing frequency and how do you carry out stability studies, here it differs a little from ICH. It says that for your products, normally you do what is called as long-term studies, and long-term studies are done at 30 degrees Celsius, which is considered at ambient temperature, plus minus 2 for fluctuations with a 60% relative humidity and for a period of 12 months. So you check the content with respect to assay, as well as when you want to see the accelerated studies, you do it at a 10 degrees higher, like a 40 degrees Celsius, about 2% variation, but the relative humidity percentage rises by 15%, that is 75%, and that decreases the tenure to from 1 year to 6 months. Now this long term studies can be done for a drug with the proposed life. If you are doing it for a drug whose proposed shelf life is 1 year, you have to see that you have to sample it for at least 6 months for first year, and then and second year, and then annually at least once in a year you pick up the sample till you complete the proposed retest period. At the same time, when you are doing or planning accelerated stability studies, you have to put it at least 3 time points, which include your final time points, that is your 0, 3 months and 6 months, to form a 6-month studies. Now this is for the drug or the dosage form which is stored as ambient temperature. There are some drugs which are to be stored in refrigerator. For that you will carry out long term stability studies at a refrigerated or refrigerator temperature. So in that case maybe 8 degrees Celsius plus minus 2 at 60% relative humidity, and accelerated should be 10 more degrees. So if you are doing long term at 8, accelerated would be at 18 degrees Celsius with the same appropriate relative humidity condition. So these are the few guidelines about it. Now how do you say it is stable or not stable? So the AYUSH guidelines, especially the CCRS guidelines, say that it is considered to be unstable if there are significant changes. Now, or if it is said to be stable if there are no significant changes. So what are the significant changes? The significant changes include a plus minus 20% change from the initial value. So if you are using a marker compound, say for example for cinchona I am using meconic acid as my marker compound. So if my meconic acid content increases or decreases by 20%, I said there is a significant change. Similarly, if my active compound, so digitoxin and digitalis, changes by plus minus 15%, I say there is a significant change. Not only that, when you do a TLC fingerprinting, either appearance of a new spot during a storage tenure, what that indicates? That indicates a new metabolite formation, or deterioration, that means degradation or vanishing of an existing spot. So if you have a new spot in TLC, or if an existing spot in TLC vanishes, still you say it is a significant change. Not only that, your physicochemical parameters as given, you should say that not more than 10% moisture content. If the moisture content, ash value, particle size, all of this vary beyond 25%, still it is considered as significant change and the compound or the preparation is rendered as unstable. So not only that, for the individual drugs, if it is not meeting the what you call it as the pharmacopoeial monograph criteria, causing a substantial change in physical, chemical or functional attributes such as your color, even phase separation for the product, caking or even hardness, change in the tablet hardness over a period of time, those all of them are also considered as significant changes, and then you will have to reformulate the drug because it is considered as unstable. So in conclusion, we have your pharmacopoeias which give you the guidelines, you have books, you have references, you have WHO standards which will give you details of all the quality control tests, ICH guidelines for carrying out different type of studies, as well as CCRAS guidelines for stability studies as well as quality control of Ayurvedic preparations. So here are few references to all of this. You can even have the document access from here, and thank you everyone for your patient listening. Thank you.